I'm going to talk about the challenges of managing heterogeneity. We've heard a little bit about that today, and it's the story that weaves throughout all of our difficulties in the rare disease space. And I want to talk about the story that brought me into that realization, and then some things we're doing to trying to fix it, and things that we can do and maybe a Center of Excellence could do. One thing I try to put forth to people that we are in the golden age of rare disease treatment. There's so much happening right now since this time, which was 1998 in the trial we did, but the last 20 years there's so much coming forward, and there's so many technologies coming forward. So many different parts of the whole cycle of information, and that means there's so much complexity on the drugs and what they are, and then on the diseases, and that means we're going to have to think very hard, and we think it's one of the reasons that we need more experience in rare, but really good expertise and experience in rare is relatively rare. There's not that many people who have been going at it for 20 years. And the development is so different, and there's so many other aspects of this that are just unique. It's extremely important that we get really good at this. And while I appreciate it's not the clinical review exactly, what there is an area, sort of the methodologic part of what we do in the rare disease design and conduct of studies that is unique and might be benefited within a Center of Excellence rather than clinical review exactly. It's a little bit more methodological innovation and helping us develop better methods for managing and conducting quality clinical development programs and data sets to help us establish safety and efficacy for our drugs. So the largest problem in translating rare disease treatments is heterogeneity, and this is probably the first word you guys in the back can read, right? Heterogeneity, I put it in big. I thought of changing all the letters, but to show how variable it is. Heterogeneity is just incredibly complex in the rare disease space, and it really distinguishes rare disease things because there's so much genetic type heterogeneity, but then there's period of diagnosis, the age, the progression, the distribution of disease within patients is heterogeneous. And we heard a little bit on the amyloidosis story of heterogeneity, and we're trying to put people into buckets, but they're not in buckets, and then we kind of miss the opportunity to actually learn and get it done right. And we need to do better on this, and so we'll talk a little about some examples. The hard problem then is that with small numbers of patients, you can't power your way out of it. You can't select and power your way through the solution to that problem. And so we need to get better at managing heterogeneity and work on the paradigm. Now, the development paradigm is deeply ingrained in us, which is the single population and a single primary endpoint and a randomized study. But that paradigm has a very simplistic question and answer that doesn't really fit what we're doing in a lot of complex rare diseases that affect multiple systems. And so we kind of need to think, how do we adapt that development paradigm so it fits in situations where you cannot screen and select varying patients, you cannot overpower your way through problems, and you need to think harder about how to deal with all these aspects of heterogeneity and how we get through this and actually learn what our drugs are doing in an effective and rigorous way. So today I want to talk about three areas, which is selection of populations, single endpoints, and trying to assess complex disease, and multiplicity adjustments, one of my pet peeves in the rare disease space. And I'll go through this and try to give you some examples and lead you through the story of Aldurazyme and some other examples to kind of capture how we can go off the rails on what we thought was traditional and appropriate, but doesn't always lead to good scientifically correct answers. So selection of patients for study, I showed this picture before. These are all MPS I patients. I show this picture because this patient here is Hurler. This is Shay, and these are various people in various stages of their lives with MPS I. The important thing is the difference between the Hurler and the Shay is that one has 99.5% deficiency and one has 100% deficiency. The Shay patient has 0.5% of normal, and that distinguishes the phenotype. A very subtle amount of enzyme, and suddenly the variation in phenotype is dramatic. And within a patient, you can see great variations in what's manifestations. Here's an old patient who has tremendous and large liver. This patient had terrible problems. His liver was not very big. In fact, it was below normal. So how do we understand liver size, or how do we understand any endpoint when patients are so variable? Now, this was the first study we did. When we looked at the phase 3 study, and this is from, I'll show some slides that came from the advisory committee, this is the population that was a selected population who had impairment in respiratory function and who had to be able to walk. And still the variation was just phenomenal in terms of what patients had. And each patient had their own particular mix and match of symptoms and problems, and creates tremendous problems. This is the first study, and so we're learning as we're going. Now, the challenge in this study, I won't go through the whole design because we don't have time, but it was a randomized controlled phase 3 study, and 6-minute walk test was one of 2 co-primary endpoints. The other was pulmonary function. We selected for patients who had pulmonary impairment, but we couldn't select for both because we had that, we'd have lost 2/3 of the patients who would never be able to enroll the study. So these are all comers essentially on the 6-minute walk. They had to walk, but not a particular amount. What's important to recognize here is that, first of all, I just want to touch on the age. The age ranged from 6 to 43 years. 6-year-old children and 43-year-old middle-aged adults are in the same study, and it's only 45 patients. So that already tells you, oh my God, these are patients at all different spectrum of disease and age. And then look at the walk distance range. 14 is the lowest, 591. So a 500+ meter range in walk distance at baseline. So the range was 10 times the minimum important difference of 54 meters. The range, heterogeneity, is 10 times the expected change. So most of you would have said, well, this is impossible. There's no way this is going to work, right? This is like, nightmare. And we unblinded this, and the blind team said, oh my God, it worked. Look, the treated group went up, the placebo group went down, but it was 0.066. But look at the pattern, it's biological. And in fact, when you went later and looked at extension, that these patients continued to get better, then the placebos that went down started getting better. A very nice effect, we thought. Now, if you use covariates for age and baseline, we actually got statistical significance that was pre-specified. However, the way it was analyzed with this tremendous heterogeneity is that the conclusion made then that we missed the endpoint. And in fact, at the AdCom, the discussion was we need to do another study, that this was not sufficient. And yet you look at all the pattern and you know that there is more to this. And with that much heterogeneity, to get even close tells you the drug has to be doing a lot. And we missed, by not looking at a number of things, we actually missed our opportunity to get the right answer. As another example of challenges in the same study, we didn't select for shoulder flexion or sleep apnea. We did not select for those 2 things. The shoulder flexion and sleep apnea both missed as secondaries. They were trend, but they missed, because only half the patients actually had shoulder flexion impairment. The other half didn't. If you look at the half that had shoulder flexion impairment, you can see the placebo and drug again separated and was statistically significant, this half. And sleep apnea, same thing. If you check people who had sleep apnea at baseline, they got better. But if you mix them with people who didn't have the problem, then you diluted the effect and you lost it. But because of this sort of religious focus on ITT is the only way to analyze everything, that we end up missing an opportunity to understand what's really going on here. The truth is, if shoulder or sleep apnea existed, they got better. So the problem here in all of this is that we're using statistical approaches that are not optimal for variable populations. The use of ITT in an inappropriate way. This is not the purpose of ITT. And when we have heterogeneity, we won't get the right answer. Time course and time integration from repeated measures is an extremely powerful way to verify that the difference you're looking at is not a random one-off thing. And when we have multiple endpoints showing the same pattern of separation, that shouldn't be considered like independent things. Those are actually feed on each other. It tells you there's a pattern of efficacy here, the totality of data thing. Now, at the time, the flaws of heterogeneity were there, but we felt we certainly had efficacy, and ultimately the AdCom voted 12-0 on both endpoints as being significant, and the drug was approved. But we also presented something else, another way of looking at this, the multi-domain responder index. And I want to put this forth because I think this is the way we should be moving. This is the method that will solve the problem we heard from amyloidosis or from anything. And what this says is I'm not going to judge what each patient might have or what their situation is, but we're going to look at the 5 or 6 things we think are clinically important, and we're going to create what's an important positive result or negative result, what that threshold effect is. And we're going to look for big wins or big losses in patients. And we'll look at the cloud of patients and where are they moving. and we'll figure out which endpoints are showing. And you don't have to then guess what's the right particular endpoint, how do you particularly do it, you don't have all that. So we did an analysis of this Aldurazyme phase 3, those data I just showed you, and we used MIDs established from other diseases. And I like to clarify for FDA, we'll never get MIDs validated for our disease because there's never been done and it'll never be done. So you have to use MID information from variable diseases and determine how well do they fit. And because of that, and because it doesn't matter, if you don't have the problem, you don't score on this system. It only picks up those that have a problem and if they change. And so the MID acts as a filter for just random variation. Only big moves up or big moves down are actually what you're looking for. So we took that information, we learned a lot and put forth a new study for MPS 7, which has even more rare disease problems, how to reduce randomization bias and enhance power measuring all. We used a blind start design, which I briefly mentioned. We used a biomarker, which still not qualified as primary in the US, but helps you understand that all the patients are actually seeing an effect. And then finally, looking at the MDRI analysis, we put that in as a clinical endpoint for the program. And we're dealing with a disease that's MPS 7, 200 patients in the world. Now this seems impossible, and that's why we did it, because it seems impossible. And we're going to run a blinded study in 12 patients. And we're going to use these techniques now we've learned and apply them in this particular disease situation. The blind start design takes patients and randomized to 4 groups. The key is that everyone gets treated and the assessment is before and after. The patients just don't know which one's their baseline and which one's their after, and therefore you eliminate or you change the pattern of knowledge, plus you gain some control on treated period information. It turns out when you model this, which we did, it's 5x more powerful than a randomized control study of the same size with the same data, 5-fold. And the problem is you don't have randomization bias, cause the heterogeneity thing, causing problems, in particularly how the study happened to randomize any particular way. It eliminates the randomization bias issue and gives you twice as much data as well. So we ran the study this way. Of course, the biomarker tells us that we're having an excellent effect on the biomarker in these 12 patients, right? Powerful, very deep, as good or better than other MPS enzyme therapies. Now, the MDRI tells us what's going on clinically. And what you look here is blue is positive, yellow is negative, gray is no change, black was not accessible because it turned out some patients couldn't do things, but that's okay, it doesn't mess up the study. Only 2 patients or 3 patients could do the pulmonary function testing for some reason. We didn't know this, they just couldn't do it. It doesn't matter, it falls away. Shoulder flexion, we assumed, since other patients had restriction, they would have restriction, but it turned out none of the patients had restriction, so nothing's happening in shoulder flexion, but it doesn't ruin the study. And we had movement in the physical mobility, in their a little bit in visual acuity, and 6-minute walk tests were contributing to what was a positive result, in my view, a half domain average improvement in p-value. Now to get to a p-value anywhere close with 12 patients, right, 6 domains, is a miracle, right? So let's pause and note the miracle. And we were very pleased. I think what's the other way of looking at this data is that there's a 3-to-1 ratio of improved to worse domains. And so I think as you look through that data and through the control data, you can get a feel for the improvement. And the agency did approve MIP-7 last November. So the MDRI has a lot of advantage. I actually don't have time to go through all of it, but the MDRI is a different way of capturing benefit where it exists in multiple domains. You learn more about the disease, and it allows you to do an all-comer study. An all-comer study, what a difference that would make if we just enroll everyone and evaluate what we can evaluate on each person. We get safety information on everyone, and we gain power from those who can measure things. It's another way of going about this development in rare disease, and would help alleviate a lot of the crises that are existing. We'd learn more, we get better data, we enroll faster, and we'd end up going to market with a better understanding of what this is going to look like. And the MDRI allows us to do that. That's why we think it's so powerful. I want to touch on one last thing very quickly, multiplicity. I'm a very negative proponent on multiplicity. I think it's way overused, and it really is not being used in the way it really is intended. It's designed really to avoid fishing expeditions, where someone does a rare disease study and they have like 30 endpoints, and then 2 things show up, and then we say, oh, look, it worked. Well, that's obviously not true. It didn't work. It just, the multiplicity would tell you that just randomly you're going to get, something 5% of them coming up. But many times the anti-multiplicity attitude kind of fails to look at the science in front of you and make a good decision of what the experiment is. And we need to find a balance between what I'd call statistical purity, religious statistical purity, and getting the science right. And I'll show you some examples of how that gets crazy. The first example is one of the craziest ones ever. This is a non-clinical study, and we're looking at GAG levels in tissues. And we had 9 tissues, and all of them were less than 0.02 and improved in this treatment. And the reviewer said, well, if you did multiplicity with Bonferroni, none of them would be positive. So what he meant was, if I only studied 3 tissues instead of doing more of a comprehensive study, and they were positive, then we'd have a real result. But because I did 9 tissues, and I had to do Bonferroni, which of course is extremely conservative, but there's something incredibly silly about that, right? Does that mean we should be doing less evaluations in our study to get significance? Are we scientists? Are we statisticians? What are we doing? The truth is, you shouldn't do multiplicity adjustment here, except in the situation you did 20 and nothing hit except 1. Well, then multiplicity becomes a problem. But if you do 20 and they hit, or 9, and they give you the answer, this is more rigorous science. But this is kind of an example how it can get to be, like, multiplicity seems to be what I'd call the sort of statistical story du jour craziness. So let me talk about the icatibant phase 3, because it was an example how heterogeneity crushed a study by accident that was a good study. This is a product that treats hereditary angioedema. They had a phase 2, phase 3-like study basically that was treating icatibant against tranexamic acid, which was another treatment, which showed very good results. And they did another one for a placebo because they originally wanted a placebo study. And oddly, they missed the primary endpoint, 0.142. But the weirdest of all things is that the 3 secondaries actually hit. And the primary was a combination of the 3 secondaries. But the interpretation was, you missed your primary analysis study, failed, which is the statistically pure thing. I would have said, well, what the hell's going on if the 3 secondaries hit and the primary is made of the 3 secondaries? Well, it turned out, of course, what's the answer? Heterogeneity. Within the secondaries, cutaneous swelling, which is one of the symptoms, and abdominal pain, which is another domain of problems, their average resolution time on placebo was 3 hours in this and 10 hours in the other one. They had different time scales. And then they mixed time. In the primary endpoint, the time was 4.6 was the average, but it had a bimodal distribution. So of course it failed. It was not normally distributed. This shouldn't have been accepted, because it was like mixing apples and oranges statistically. But if you just look at this, you'd say, this is weird, first off, because you have a great study here and have this weird thing happen. But if noticing the heterogeneity would have been an issue. Now, what I'll show you, if you look at, this is from the EMA review, this table. If you look at all the purple, which is 0.05, and yellow, 0.10, you see that almost all the stuff are either significant or trended. So forget about purity and multiplicity. They made an accidental guess of how to combine them. It was wrong, that guess. The drug is clearly working. And most importantly, we talk about patient focus, the patient's view of their symptom resolution was 1 hour on drug versus 17 hours on placebo. 1 hour versus 17, and this was 0.01. This is the patient view, and the doctor view was similarly extreme. But this study was felt to fail, and the question asked, well, what happened then? And well, the EMA actually approved it in this case, but the agency didn't, and it ended up getting approved in a second phase 3 where they didn't make the same mistake on the statistics. It got approved, but the problem was that this company, Jerini, stock crashed without the US approval. They got sold, parted out, the product remained, and then everything else, the technology on peptide modeling technology, which they had, which was very cool, lost. And so the whole thing got blown up because of heterogeneity. So a very important example of how the unintended collateral damage, if you will, of the decision. Last example I just want to touch on, which is a more recent one and a good one, and how it can be done differently, is the beresimab story, and which is called Crysvita. This is for a treatment for XLH. This is a patient, adult patient with XLH. Obviously, it's not a mild disease for some people. They're mostly bionic now. So in this case, I won't go through a whole study design, but it was a randomized control study, and we had 3 secondary endpoints: pain, stiffness, and physical function. And then we pre-specified multiplicity adjustment, which I hated doing because I just think these are highly related to each other, by the way. So they're not true independent shots on goal, but we're going to act like they are. So when we did the result, you can see the pattern of data, but stiffness was significant and WOMAC nominally, but with multiplicity, it said stiffness was the only one. But if you look at placebo and drug in all 3, you can see clearly all 3 are moving in the right way. And at 48 weeks, the crossover patients crossover, everyone's getting better. So the multiplicity approach says only one's positive, but the scientific view of this would say that actually all of them are clearly moving. Now, importantly, because of multiplicity, we put the fracture endpoint in the alternative exploratory, because we didn't want to hurt the multiplicity of this thing. And we didn't know how many patients would have fractures because of heterogeneity. And you couldn't know for sure at the moment of baseline who would have a fracture or who wouldn't. It turned out half the patients had fractures, and our exploratory endpoint, of course, should have been the one that was secondary. The p-value is 0.001. The odds ratio was 17-fold for being fully healed on drug versus placebo. 17-fold, so that's huge, right? To the PhD credit, Dr. Kehoe and the team, they said, wow, this is amazing. Yeah, I see this. This is amazing. It is amazing. And they ignored the issue of where it was. They said it's amazing and we're moving forward. That ends up in the label and approved. And I think that's right. The science was there, but the random guessing and choicing and the multiplicity in the game we have to do sometimes has to be overtaken by common sense scientific view of what's happening. And that did happen. That's a great example. What could a center of excellence do? I think what I would look at it as more support for new methodology and advancing innovation, where a division doesn't have time to look at a new trial design, like do the modeling of it or whatever. They don't have time to figure out some of the methodological aspects of things, and therefore they can provide that support of, is this thing being proposed actually sensible or not, and help with innovation and adoption of new methods, new study designs, and other things. She mentioned there's another group now looking at qualifying biomarkers, which is great. But we want to make sure that there's a group perhaps enhancing the method development that's helped and be able to advise the different rare disease divisions of the clinical knowledge that she's talking about in those more specialized divisions. So heterogeneity issues for rare disease will be always the thing. So it's a golden age, but managing heterogeneity is key. We need to come up with better ways of doing this, and including, I think, things like all-comer studies, MDRIs, biomarkers, and being really good at understanding how to manage heterogeneity will make us more successful. Thank you.